Let us talk about biological molecules, particularly carbohydrates and lipids today. So, if we think about the basic categories or groups of families of biological molecules, we have already talked about proteins and nucleic acids. Carbohydrates and lipids are the other two biological molecule families that are very much abundant in living systems. So let us talk about carbohydrates first. Carbohydrates are one of the most abundantly occurring organic molecules. So they are one of the major dietary sources of energy, and they are mainly made up of carbon, hydrogen and oxygen. So this in carbohydrate also what we have is basically monosaccharides, which are the repeat units of the polymer, and they basically have a general formula of C6H12O6, and this is something that you all are familiar with. So saccharides in general can be classified into three groups based on the number of sugar units they have. Single unit will be called a monosaccharide, oligosaccharide or polysaccharides. Okay, monosaccharide is a single unit, oligo and polysaccharide, we will talk about them as we discuss further. The first two, that is monosaccharide and oligosaccharides, are crystalline in nature, while polysaccharides might not be. So monosaccharides and oligosaccharides are generally sweet in taste, they taste sweet, they are water soluble, and they are crystalline in structure. Now carbohydrates are precursors to several organic compounds as well. They store form of energy to meet the need of the body, and that is why we say that when you suddenly feel tired, we immediately drink glucose, because that is something that most easily can get converted to energy. So let us talk about monosaccharides. Monosaccharides are the simplest group of carbohydrates, and they have in general the formula of CnH2ON. So when it is C6H12O6, this N becomes 6, right? They are further divided into two types. One is called aldose and another one is called ketose. So in aldoses, the functional group will be an aldehyde group, and for example like glucose, and for ketoses the functional group will be a ketone group, which is more familiarly found in case of fructose. So on the basis of number of carbon atoms, monosaccharides can be referred as triose, terose. So only a few, like a single or a couple monosaccharides could be there. In case of oligosaccharides, you have 2 to 10 molecules of monosaccharides, right? So they can be divided further divided into classes like disaccharide, trisaccharide and so on. But monosaccharides are referred to trioses, teroses, pentoses, hexoses, depending on how many carbons you have. That is the number of N that you have, right? CnH2ON. So depending on the number of N, it could be called triose, terose, pentose, hexose, heptose, like that, depending on it is 3, 4, 5, 6 or 7. For example, glucose will be called an aldohexose. Aldohexose. Why? Because C6, that is why hexose, and because the functional group is an aldehyde group, that's why it is called a aldohexose, right? So this is how you name them. Now let us discuss further about disaccharides. This is one of the most common oligosaccharides, where you have two monosaccharides sitting side by side. These names also I am sure that you are familiar with, like maltose. So maltose is basically where you have two glucose units, isomaltose, silobiose, sucrose. Sucrose is another familiar name that I am sure that you know. So these are all disaccharides. That means they have two molecules of monosaccharide forming one oligosaccharide. So second in the family comes lipids. Lipids are structurally most diverse between these four families. By four families I mean proteins, nucleic acids, lipids and carbohydrates. So we have already discussed 3 of them. Now let us talk about lipids. So lipids are structurally most diverse, and this is because that unlike the other members who have a common kind of a common structure, right? So we have talked about nucleic acids or proteins or lipids. Everywhere we saw that the monomers were very much clear that what my monomers were. They could be 1 or 2 or 20 separate structures, but that's it, and then you can organize them in different order to get the polymers. In case of lipids, rather, there is not some clear structural feature, right? So there is no clear structural feature, so to say, but what you have is a common physical property that lipids are all hydrophobic. In general, lipids are going to be hydrophobic. So let us see that what kind of properties we mean by this. So hydrophobicity, as you already know, that is afraid of water. So anything that you see around us which are in general not very fond of waters and they belong to a biomaterial, that is most probably having a large lipid component. For example, all the fatty acids, wax like that use, wax does not mean only the mombatti kind of thing that we use, but there are different kind of waxes, and all of them are basically combinations of fatty acids and alcohols. Triglycerides, in general you have 3 fatty acids and a glycerol molecule. There are steroids, which are basically cholesterol derivatives, and above all there are membranes, and we'll talk about these membranes in little details. So these membranes are basically, whenever you see any biological entity, it is usually covered by a certain membrane, a certain separating unit that separates the inside from the outside. Starting from cell membrane, everywhere you see these layers that separate out inside from the outside, and they basically are made of fatty acids. So in general, fatty acids contain a carboxylic group, that means a COOH group, and that should make them quite polar. However, in general, and polar means they should not be very much hydrophobic. Polar means that they should be fond of water. But in general, what they also have is a very long hydrocarbon tail. So if you see that this is a structure of a fatty acid, in general you have a COOH carboxylic group, right? And it is an acid group, so that means acids are usually soluble in water, and you know that. So in general, COOH group means that it should be fond of water, it should be having a polarity, and that it has. Its heads are basically fond of water, but on the other hand it has a long non-polar tail, and this long non-polar tail is usually made of some hydrocarbons. Here you can see a long tail like CH3, CH2, 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 long tail. This entire tail is non-polar, and this non-polar tail is basically responsible for the hydrophobicity of fatty acids. So it basically, you can imagine it to be a structure where you have a head which is hydrophilic but the long tail which is hydrophobic. So these saturated fatty acids usually depend on their melting points, their properties, physical properties. They depend on how long that particular tail is, how many carbons are there in that particular tail. Here in one graph we are showing that the melting point of these fatty acids are basically rising depending on how many carbons you have in the molecule. So different kind of structures could be there, and the common trait that you will find in all these structure is always this head, COH. This head is a polar head, and with that you have this long tail which is a non-polar tail. So that is the common structure of any fatty acid. So as you know that acids, when react to bases, they basically create a salt and water. Same thing is true here also. Here also, if you make the fatty acids react with NaOH like bases, then you will get a salt, and along with that you will have water. So this is the salt, and you will have water present in there. So now let us talk about that how these fatty acids can create something like a membrane. What is the importance of these fatty acids in making a membrane? So the basic structure you can imagine it to be that a polar head and a non-polar tail. That is what we have learned so far. So the salts of the fatty acids that we have just described in the previous slide are called soaps. These are basically soaps, and they are called that they have a particular property that we call them, they are considered to be amphipathic. Amphipathic means they have a part which is very hydrophobic along with a part which is very hydrophilic, right? So this is the property of a amphipathic material like soaps. So when placed in water, what happens is these molecules can form beautiful structures because of this particular very important property, that one part is very much hydrophobic and another part is very much hydrophilic. For example, they can get combined in a form like this. If you look into this form, what we have here is basically, it's a three-dimensional structure like a ball, where all the parts, all the heads are assembled together, saving their tails inside so that no water can penetrate inside this region. So if this blob closes, if you imagine a 3D ball and the outside of the ball is all made of these heads of the fatty acids and tails are all inside, then they are safe from water. So these parts, the tails, are hydrophobic, so they are safe inside water, while the hydrophilic tails are hydrophilic heads are closely lying in water. These kind of structures are called micelles. When they organize in this form, let's suppose this way you have water, right? Suppose this is your water, and on top you don't have any water, so just on the water surface they will be arranged in form of a layer, and this layer is called a monolayer. Just like the monolayer, bilayers are basically what we see in most of the biological membranes. So biological membranes are these selective barriers to movement. You can commonly find them in cellular structures in any living cell in form of the cell membranes. So about cell membranes, we have learned so many different things before in our school days, that they are selectively permeable, only some materials pass through them and some cannot. They have something called active transport, passive transport. Let us talk about these things today, and let us learn about the structures also. So in biomembranes, usually what you have is some part of carbohydrates and lipids, both present sometimes, but lipids are the main reasons how these are made. Sometimes cholesterols are also present there. So let us find out the common structure of these membranes. So as we have discussed that fatty acids have this head which is polar and a tail which is non-polar. So what happens in case of these membranes is usually what we have in most of the membranes is something which is called a phospholipid. So in phospholipid you have a phosphate head and two tails, similar hydrocarbon tails, two tails made of fatty acids. So a head that is extremely polar and two tails which are non-polar. And so this hydrophilic part, the head, is always attracted to get in contact with water, while the hydrophobic part, that is the tail, they want to be away from water and they want to be protected from water. So and thus they create these things which we call bilayers. So bilayers are basically when you can form two layers with these phospholipids to create a structure where the tails will be completely protected and you can enclose a particular volume with this kind of a layer. So this is the very initial model of how these structures of cell membranes are made, and this is called Danieli-Davson's model of cell membrane. So what you have here is that you have these phospholipids oriented in a very particular structure. So let me show you another picture which will be much more evident. So here you can see that in this particular picture, suppose this is the extracellular fluid and this side is the internal parts of the cell, and this is the cell membrane that separates the external and the internal part. So this is your cytoplasm and this is your extracellular fluid, outside cellular fluid, and they should be, both of them have a huge water content. So both of them have almost 60% of water. So that means the tails that we have in this phospholipids, see, black portions are the tails. So these tails, they don't want to be in contact of that. So for this reason, the hydrophilic heads, so this is the part that is hydrophobic, right? So hydrophobic zone is enclosed inside, while the hydrophilic heads protect them from outside. So these are the hydrophilic heads. This is also hydrophobic heads. But inside you have the tails which are hydrophobic, they don't want to be in contact of water. Thus you form a bilayer, a layer of two layers of phospholipid which are sitting side by side, and inside they are protecting the hydrophobic tails. So this is basically the basic structure of phospholipids, and that can be verified once the electron microscope was invented. It was seen that what you have as a cell membrane is basically having two layers, right? When you study them under microscope, you see that they have two layers. But sometimes it is also found that parts of it have some other molecules present as well. It was found through a particular technique which is called freeze fracturing. In freeze fracturing, what they do is that basically it's a specialized way to split the membrane from the middle of the lipid layer. So you have two lipid layers, you try to split them, you freeze them such that they break down from between. So this is something like this, you freeze them and you then split it open to see that is there only two layers. So you can see this in very evident way. These are all the heads. These are all the heads that you have here. The tails, the yellow portions are the tails and the gray portions are the heads. So if you use this technique, which is called freeze fracture technique, then you will see that the inside and the outside is basically constituted of heads only. But if you open it up, sometimes you can see some proteins also sitting here. So the structure is not just a bilayer, there are some other proteins also protruding here and there. What is the purpose of this protein? They have several different purposes. Because the internal part of this cell membrane is hydrophobic, so nothing that contains any amount of water basically can pass through this membrane. But that should not be there, right? The cell needs water, the cell needs food. So several different water containing stuff should penetrate inside. So these proteins basically manage that transport phenomena across the cell membrane. So if we look at the structure of these membranes, they are not just bilayers. Bilayers is one of the major constituent of it. They are the membrane that separates, but in between you have some peripheral proteins which are bound to the surface of the membrane, and some integral proteins that are permeating the membrane. They are exposed on both sides. So if you see this picture, some proteins could be can be seen from outside only, and some could be penetrating the entire membrane sitting on both sides. Their ends protruding out from both sides. So they are called integral protein and peripheral proteins, and the final structure is called a fluid mosaic model. So it seems as if the membrane lipid layer is basically like a fluid, and in that fluid the proteins are moving around, floating around side by side. So the structure is called a fluid mosaic model here, where we have this part where the membrane was formed with the heads outside, tails inside. But to make other quantities like water, sodium ion, calcium ion, potassium ion, other important materials that the cell needs or the cell throws out, to get those stuff in and out, some proteins are sitting on the cell membrane as if in a mosaic pattern. So as if in this fluid you have proteins here and there, and they are floating around. They're floating around and they're doing several different stuff. They also sometimes you have parts of the molecules, proteins and other molecules who are there to sense the outside temperature, maybe the outside chemical gradient maybe. So these things are required on the membrane, right? So sometimes the cell also includes those parts. So the integral proteins would be when the both sides are protruding out on both sides of the membrane, and the Peripheral proteins will be when they are only sitting on one side. Their side is only visible from one side. Other side, they are not visible from the other side. So then it will be called a peripheral protein. And there are non-polar amino acids. These are non-polar amino acids. That's why they can sit with the hydrophobic tails. No problem. They are the non-polar sides. This is the non-polar part. So they can sit with the hydrophobic part. No problem of them sitting with the hydrophobic part of the proteins. On the other hand, the polar amino acids, which love water, and they can protrude outside, like this part and that part, they can protrude outside. This is the polar amino acids of the proteins. We discussed about how the amino acids could be polar or non-polar. So this is how they orient themselves. So this is the general structure. So as we have discussed, hydrophobic heads, hydrophilic tails. Just remember this basic structure, the two tails of the phospholipid and the fluid mosaic model. So this is the total fluid mosaic model, how the membranes look like in general. There are cholesterol molecules also who sometimes work as sensors, and this is how one system. So why we are talking about membranes in so detail? Because this is how one system is made with different parts of the system getting created from different types of biomolecules, right? So the biomolecules, all of them are different types of polymers. You know now that the proteins are also polymers, right? You know now that this lipid bilayers is also kind of a polymer like structure. They are sitting side by side, a kind of arrangement, right? On the inside, the lipids themselves is also looks like a polymer in the portion where they have their tail. So how this structure gradually grows from a single molecule to a complicated molecule to a complicated system, and finally a totally functional part of our cells. That is how it is done in every part of the living beings. This is how the biomolecules are constantly, biomaterials are constantly, layer after layer, they have this hierarchical structure to evolve from a single molecule to a complicated system. So we can call these membranes as a system because it's a fully functional machine. It covers a particular part of the that it is supposed to cover. It makes sure that whatever it does not want inside does not get inside. It makes sure that whatever it wants inside or outside, that has its way to go inside and outside. So you can call it a machine, right? So that is how from the single biomolecules gradually by self-organization, this is what we call self-organization. No one is making these things happen sitting there. No one is there to tweak these machines so that they function in this particular way. Because of their properties, because of their physical properties like hydrophobicity, hydrophilicity, these things are happening, and from single biomolecules, complicated systems or machines are being made inside living bodies. So we are concluding this section of polymers and biomolecules in this part. We'll start with nanobiotechnology in the next class, with a brief mention of active transport and passive transport before that. So let us stop here.